a bit behind the schedule, but the next speakers is Sara. I hope I pronounced it right. Sara Okhuijsen. Sorry, sorry. Sara is the co-founder and the CTO of the company called OASYS NOW, a Dutch health tech startup that built privacy-preserving explainable AI for clinical research and also precision medicine. She's recognized as for Forbes 30 Under 30 Europe and the Women in the AI Build looks and with additional Women in AI award. Sara will introduce this confidential multi-party AI platform they developed, a secure framework created for OASYS NOW and leveraging the confidential computing. So Sara, please take away.
Yes, thanks, Chester. Can you see my screen?
Yes.
Nice. All right. Yeah. Thanks for the great introduction. Yeah. So I'll give a talk about confidential federated learning for collaborative genomic research. It's a good program because it follows up very nicely on like Jeff's talk about collaborative research with oncology and Roche from Eric and also Juval's talk. So I'll talk about collaborative genomic research. First, I'll give a little bit of context about genomic privacy. So applying these techniques specifically to the field of genomics. Then I'll introduce the use case of genomic collaborations and the setup and assumptions that we take. We'll dive a little bit in the privacy and security threats. So actually the raw data model leakage during training that Juval also highlighted, specifically important for genomics. How do we then address those? The security framework that we've built. So first, a little bit about OASYS NOW. So we're a startup in Europe. The whole compliance part that federated learning brings is very welcome here. Also in healthcare and in genomics, the privacy benefits are obviously very there. So we make hidden health data actionable. We have products live for clinical trial recruitment and real-world evidence. What I'm going to talk about today is more R&D. So it's more about the genomic privacy niche, and that's where I also do research in. So a little bit about genomic privacy. So I just want to give you sort of a scope of what's currently out there, what frameworks are currently already exist, and and I looked at their like privacy and utility trade-off. So if you look at like genomic data as a bioinformatician or as a computational biologist, you basically get schooled about this more linear approach of like you collect your data, you process it, you analyze it, and then you publish it. And that's sort of where the education stops. While it's super valuable data that you would want to like reuse or repurpose, needing a lot of SOPs and standards that and good practices that computer scientists would know more about typically than bioinformaticians. But fortunately, we're all realizing this. What I want to do here is sort of look at this genomic data lifecycle that we need, where we need to also integrate the data back and see what privacy challenges occur on that road. So for integrating and processing data, a typical privacy challenges that arises is secure outsourcing. Hospitals, researchers, they might not trust a third party or a cloud provider. There's just an inherent risk of data exposure. Also, another challenge is secure collaborative computation. So you want to work together across institutions, but you might not want to centralize the data. In genomics, also, output privacy arises as quite a big challenge. So if you want to publish about your research and you publish even summary statistics of your genetic data set, you might be able to deduce which person is in this data set, giving them maybe risks of future employment, insurance, etc. Storing and finding, sharing, they have similar challenges. Secure querying, for example, in sharing, in finding, and secure sharing. with OASYS Now, we want to work towards an end-to-end platform for genomics, an end-to-end confidential platform for genomics. In this talk, we'll focus mostly on the challenge of secure collaborative computing. So that's what we'll zoom in on. But yeah, good news is that there are already a lot of there's a lot of good research going on. Here's just a list of a bunch of frameworks that are out there and what technology they are using: homomorphic encryption, secure multi-party computation, differential privacy, or a combination of those. So yeah, it's a very thriving field, so to say. So yeah, we're zooming in on the secure collaborative part of the problem. We used to be able to aggregate our outcomes in a more linear way. So genome-wide association studies is what researchers used to typically do, which is linear correlations between the DNA and the phenotype, so the disease. But yeah, then you can only detect linear correlations, and biology is not just linear, it's more complex. So if you want to detect those more complex relations, you need to move to deep learning. And you need a lot of data, you need to centralize your data. So yeah, the need for this using deep learning is quite clear in this field. The framework that we chose to test with is called GenNet. It's an interpretable deep learning network. So that means as an input, you have your genetic data, and as an output, you have your prediction. So based on your genome, do you have blue eyes? Do you have schizophrenia? And it's fully interpretable. So all the connections actually represent a biological connection. So the SNPs are parts of certain genes, the genes are part of certain pathways, and this knowledge is publicly available of biological databases. So they're fully integrated into these interpretable networks that you can configure, and the resulting model weights thus represent how much did this gene or pathway contribute to this prediction. So yeah, I find this a super interesting model. And that's the one that we chose to test with in collaboration with this Dutch hospital, the Erasmus Medical Center. So yeah, we assume that the researchers, they want to collaborate on this sensitive genomic data using AI. They cannot move their data off premise. They cannot move it off institution, not across borders, and out of EU, it's getting more complicated. So therefore, we use federated learning. We see in practice with implementing federated learning, researchers, more schooled like bioinformaticians find it hard to implement it securely. Legal teams have a hard time verifying the compliance, and IT departments are also a challenge. But yeah, there's a lot of good developments going on as discussed in the previous talk. So that's very encouraging to see. Other assumptions that we made that the clients, so the hospitals collaborating, are honest but curious. So they won't be attacking each other, but if their data lands on their desk, they will definitely take a look. But the clients do not trust the model orchestrator. This is typically a third party or a cloud provider. So there we did assume an adversarial setting. So yeah, the privacy threat that then arises in this setup is the largest threat is at the side of the model orchestrator. Juval already highlighted it very well. So during the training phase, the data leakage is the biggest risk. Because if you can see the model weights, you can reconstruct the original data, and for genetic data, that's just a very serious issue. So there are different ways we can go about mitigating that risk. And for that, I just wanted to take you along on a little landscape of the privacy enhancing technologies and how they relate specifically to genomic data and the privacy utility trade-off specifically to genomic data. So the first one, maybe the odd one out, is differential privacy. So introducing noise. Introducing noise to the model weights. It's such expensive data to gather. It's so hard to get all the nucleotides, and then to introduce noise. Like if I would ask a biologist that, I wouldn't really want to introduce noise to my already noisy biological data, but but also from a more first principle thinking, it doesn't really make sense for the genomics use case, because you would already be able to identify a person from 30 to 80 SNPs, so the genomic equivalent of bits, out of the billion. So yeah, you have to basically mask the entire data set if you want to truly use that as your measure. So for this specific use case, a differential privacy is probably not your choice. Then there is the software-based method, homomorphic encryption based on zero knowledge proofs. It's a truly beautiful method, but we're talking about enormous data sets, and the downside is usually that it's quite slow, or it requires a lot of optimization, but then you have to optimize the algorithms, and bioinformaticians, researchers, they tend to want to use different pipelines every time. This would be quite a downside in the trade-off. Also, it's not collaborative. So for what we're looking for, this might also not be the primary technique of choice. Then there's another software-based cryptographic technique, secure multi-party computation, also called secret shares. So this one is collaborative, but it's still like very slow. So you still get the same downside as homomorphic encryption with regards to having to optimize per algorithm. Our favorite, so to say, is confidential computing with attestation. I guess I learned from Juval that I need to specify that further than what's here on the slide. But yeah, as a broader category, trusted execution environments. So it's just incredibly fast, right? Because in the hardware, the computation happens normally, hidden inside the secure enclave. So the overhead is negligible. And if you're talking about such large data, that's really important. Typically, the downside, the trade-off would be actually the memory limitation. So yeah, you're limited by the hardware, the memory limitation in the hardware. But because the hardware has been advancing, this trade-off is basically going away. The limitation that we've been working with is like 800 gigabytes, which is barely a limitation at all. And now we're also having confidential GPUs. The implementation that we did was with confidential CPUs, but it works also with confidential GPUs. So that gives even more possibilities and options. And one small side note is also that biological data or genetic data specifically lends itself very well to parallelization, therefore also working very well with GPUs as well. This is really the most promising PET specifically for genomic data. So the solution, the route that we chose is secure aggregation. So in order to mitigate the training phase privacy leakage, to keep the model weights encrypted throughout the process, and then it's also not exposed to this party. So that's what we built. We built a framework called confidential multi-party AI, where the model weights, they stay encrypted, they get securely aggregated, and it works with this GenNet framework. It can also work with any other framework, federated framework. So the data stays on premise of the hospital. There's verifiable security against data leakage through this encrypted hardware. There's explicit trust amongst the parties through attestation. So the very critical part, you don't have to just trust each other, but you can verify it. So besides just hospitals that already work together, they can also maybe have commercial collaborations with less trusted parties, for example. And really the linkage of this attestation through to certification and to contracts. That's where we really see the key, and that's also what we hear throughout this evening. At least it's evening here. That that's the real challenge, a really practical bottleneck. So that's also my opinion, that I think we need to really see how can we bind these legal contracts to the attestation. That would really unlock a lot. And this was a collaboration with NVIDIA and with a Dutch medical center, CISPA Institute, and it was sponsored by this ELSA, European Lighthouse for Safe and Secure AI. And thanks, that's it. Ready for any questions?
Great, thank you, Sarah. Any questions? Sarah, I got a question for you. So,
Go ahead, Chester. I was just going to ask, do you include all SNPs, or do you just include the ones that were attributed to genes? And how do you do that attribution, I suppose, if that is the case?
Yeah, so that's part of the GenNet framework. So we use all the genes, and we use quite a nice data format for that. So I would definitely recommend checking out that paper. They use a very nice sort of multidimensional data format that can be used for anything, because you can have different data types also in genomics, but they used a very nice way to standardize that. So any format can be converted into that format used, and all the data can be up or down sampled, basically.
I see. No, I just mean if you're using SNPs that are outside of, like, say, the gene region, like in non-coding regions or intergenic regions, how do you attribute those to a particular gene to do this? But second, I guess the reason for the question is, like, if you were to include all the SNPs, then, depending on which set of SNPs I can query for, I can try to, like, genetic information is inherently identifying, right? So, like, I'm not saying I would try, but someone could, in theory, try to identify individuals in your data set if they can query any SNPs.
Mm-hmm.
I'm just curious how you think about that. But that's
exactly. So there's still the question after that is basically, what do you do with the published model? So then you get more to the output privacy questions. But with regards to coding versus non-coding, that depends on which framework you use. So this GenNet framework is specifically for coding DNA, but they are extending it also to non-coding. But that just depends on which framework you use.
fair enough.
All right.
Thank you.
So if there's no more questions, I probably gonna move on. So we're a little bit behind.